Okay, let's unpack this. You look at a plant, right? It seems pretty passive, just soaking up sun and water. But behind the scenes, it's doing something incredible, taking basic, inorganic stuff, minerals from the soil, gases from the air.
Yeah, just the raw ingredients.
And turning them into, well, life. Complex organic molecules, leaves, roots, everything. How on earth do they manage that?
It's definitely not passive. It's an incredibly active, really sophisticated set of processes. And that's what this deep dive is all about, nutrient assimilation in higher plants.
Nutrient assimilation, yeah.
Think of it like the plant's internal factory, taking those raw inorganic materials and building itself molecule by molecule.
Right. And our goal today, using the material you shared, is to really pull out the key ideas, the main processes, maybe some surprising bits, so you get a clear sense of how plants build themselves from, well, dirt and air.
Exactly. And a really crucial point from the get-go, highlighted in the source, is that this isn't like effortless for the plant. Not at all. For key nutrients, especially nitrogen and sulfur, converting them involves some complex biochemistry that uses a lot of energy.
How much energy are we talking?
The figures mentioned are pretty significant, about 12 to 16 ATP equivalents for every single nitrogen atom assimilated, and around 14 ATPs for sulfur.
Wow, okay. That's a serious energy investment per atom. The material even mentioned the stored energy aspect, like why things like TNT or ammonium nitrate are explosive.
Precisely. Plants are essentially packing energy into these elements to make them biologically useful. That energy can be released, sometimes dramatically.
So plants are this vital bridge, moving nutrients from those really slow geological cycles.
Like rock weathering, yeah.
Into the much faster biological cycles that, well, everything else depends on, including us, for food.
Absolutely critical. Can't overstate their importance.
So what's the plan for this deep dive? What nutrients are we covering?
We'll look at the big ones. Nitrogen, definitely. Sulfur, phosphate, various cations, things like iron, magnesium. Even oxygen assimilation gets a look in.
And that biological nitrogen fixation. The bacteria partnership.
Oh yeah, we'll definitely get into that. It's fascinating and, again, incredibly energy demanding. We'll explore just how much energy these processes really cost the plant.
Great. So let's start with the overview. What exactly is nutrient assimilation in simple terms?
Okay, simply put, it's the metabolic pathways plants use to take the inorganic ions they absorb, like nitrate, sulfate, phosphate, and incorporate them into their own essential organic compounds.
Like building blocks for...
For everything. Pigments like chlorophyll, enzyme helpers, fats, DNA, RNA, and of course all the amino acids needed for proteins.
And the material kind of splits this into two main types of assimilation processes.
Yeah, broadly. First, you have those really high energy conversions. That's mainly nitrogen and sulfur. Taking something oxidized like nitrate and reducing it, adding electrons, pumping in energy to make it part of an organic molecule. That's where you see those big ATP numbers we mentioned.
12, 16 for nitrogen, 14 for sulfur. A major biochemical effort.
A huge effort. The plant has to invest serious energy. The second type is less about chemical change and more about forming stable connections.
Like with metal ions, magnesium, potassium.
Exactly. They don't usually get chemically reduced in the same way. Instead, they form stable complexes, often through non-covalent bonds, coordination bonds, or electrostatic attractions with organic molecules.
The classic example being magnesium right in the middle of chlorophyll.
Perfect example. Or calcium helping to structure the cell wall, like in that egg box model described. It links pectin chains together.
And the function totally depends on that ion being there, right? Pull out the magnesium, chlorophyll doesn't work.
Absolutely. The material really stresses that point. The cation is integral to the function.
Okay, let's zoom in on nitrogen then. It's literally everywhere, 78% of the air. But paradoxically, it's often the nutrient that limits plant growth the most. Why is it so vital?
Nitrogen is just fundamental. It's in every amino acid, which means every protein. It's in every nucleotide, the building blocks of DNA and RNA. After carbon, hydrogen, and oxygen, it's the most abundant element in plants.
So essential for building almost everything.
Pretty much. And because usable nitrogen is often scarce in soils, adding nitrogen fertilizers is, the standard way to boost crop yields.
Right. The material shows the nitrogen cycle, how it moves between the atmosphere, soil, organisms, changing forms from N2 gas to ammonia, ammonium, nitrate, and into organic stuff. But the big hurdle is that atmospheric N2, right?
Right.
That triple bond between the nitrogen atoms is incredibly strong.
Super strong. Breaking it is called nitrogen fixation. Industrially, we have the Haber-Bosch process. High temperature, high pressure catalysts. Uses a lot of fossil fuels. Huge scale, over 100 million tons a year.
Wow. Massive industrial process. But nature does it too.
Oh, yes. Nature's been doing it for billions of years. A little bit from lightning, but the vast majority, around 90% of natural fixation, comes from microbes, biological nitrogen fixation.
And that's crucial for agriculture, especially in places where fertilizers are expensive or, have environmental costs.
Hugely important. Once it's fixed into ammonia or nitrate, it gets into the soil solution. Then it's a race, plants and soil microbes all competing fiercely for it. Plants need really efficient uptake systems.
Okay, but the material also warns about having too much nitrogen, specifically ammonium, NH4 plus area. It says it can be toxic.
It can, yeah. If it accumulates inside plant cells, ammonium can mess with the proton gradients across membranes.
Proton gradients, like the cell's batteries.
Exactly. They power transport, ATP synthesis, lots of vital stuff. Ammonium basically dissipates those gradients, short-circuiting the cell. It's why smelling salts work that ammonia hit disrupts nerve function.
So how do plants cope?
They deal with it fast. They either immediately convert ammonium into amino acids, or they shuttle any excess into the vacuole for safe storage. They don't let it build up in the main cell body.
Okay, so ammonium is handled carefully. What about nitrate, NO3? The material says plants can store that more easily.
They can. Nitrate is less disruptive and can be stored in the vacuole in quite large amounts or transported around the plant relatively safely. It's a common form plants absorb.
But there's a catch if we eat high nitrate plants.
There can be, yes. The material mentions that in livestock, or even humans, high nitrate intake can lead to nitrate formation in the body. Nitrite interferes with hemoglobin's oxygen-carrying capacity, that's methemoglobinemia.
And potentially other issues, too.
Yeah, potential links to nitrosamines, which can be carcinogenic, or nitric oxide signaling. It's why some places regulate nitrate levels in vegetables.
Good to know. So plant takes up nitrate. How does it get assimilated? The material described a two-step pathway.
Right. Step 1 happens in the cytosol, the main cell compartment. The enzyme nitrate reductase, or NR, reduces nitrate, NO3, to nitrite, NO2. This uses energy, usually from NADH or NADPH.
And that enzyme, NR, sounds important. Complex structure, needs molybdenum.
Yes, absolutely. It's a complex enzyme with multiple components, including that molybdenum cofactor. And it's a great example of micronutrient importance. If a plant lacks molybdenum, NR doesn't work, and nitrate can build up to toxic levels because it can't be processed.
And the plant controls this enzyme activity really carefully.
Very tightly. Its production is regulated by nitrate levels, light, carbohydrate status, and then there's faster control too, by chemically modifying the enzyme itself, phosphorylation, turning it on or off quickly in response to light or dark.
Okay, so nitrate becomes nitrite. But you said nitrate is toxic too.
It is. So the plant doesn't let it hang around. It's immediately transported into plastids, that's chloroplasts in leaves or other plastids in roots.
Ain't in there.
Step 2, the enzyme nitrate reductase, NR, takes over. It reduces nitrate all the way down to ammonium, NH4+. This is a big reduction step, needs 6 electrons.
Where do those electrons come from?
Usually from ferredoxin. In leaves, ferredoxin gets electrons directly from the light reactions of photosynthesis. In roots, it gets them from other metabolic pathways, like the oxidative pentose phosphate pathway.
And where does all this nitrate assimilation happen mainly? Roots or shoots?
It varies. Plants can do it in both places. The source mentions examples like cocklebur doing most assimilation in the shoot, while white lupine does more in the root. Depends on the species, nitrate availability, even environmental conditions.
Okay, so through one route or another, we've arrived at ammonium, NH4 plus estrol. We know it's toxic if it builds up. So how does the plant quickly make it safe and useful?
Right, this needs to happen immediately. The main route for incorporating this ammonium into organic molecules is called the GS-GOGAT pathway.
GS-GOGAT. Okay, what do those stand for?
GS is glutamine synthetase. It takes the ammonium and adds it onto an existing amino acid, glutamate, to make a different amino acid, glutamine.
And that step uses energy.
Yes, that's an ATP requiring step, essential input of energy there.
Okay, so GS makes glutamine. What about GOGAT?
GOGAT is glutamate synthase. It essentially takes the nitrogen that was just added to glutamine, transfers it to another molecule called 2-oxoglutarate, and produces 2 molecules of glutamate.
2 glutamates. How does that work?
Well, one glutamate is basically regenerating the molecule that GS used, and the other glutamate now carries that newly assimilated nitrogen atom. It sounds complicated, but it's a very efficient cycle.
So, net result, ammonium gets safely tucked into glutamate.
Exactly. And from glutamate and glutamine, the nitrogen can then be transferred to other carbon skeletons using enzymes called aminotransferases. That's how the plant builds up its whole suite of different amino acids.
Glutamine and glutamate are like the central entry points for nitrogen then.
They really are, the hubs of nitrogen incorporation.
And plants need to move this nitrogen around, store it sometimes. The material mentioned asparagine and glutamine being important for that.
Yes, especially asparagine. It's highlighted as being very stable and having a really high nitrogen to carbon ratio, which makes it efficient for transport and storage compared to glutamine or glutamate.
Interesting. And there's a metabolic switch, depending on energy levels.
Yeah, it's quite neat. When the plant has lots of energy, good light, plenty of sugars, it tends to run the GS-GOGAT pathway to make glutamine and glutamate, ready for building proteins and growing.
Makes sense.
But if energy is limited, say, low light or stress, the balance shifts. GS-GOGAT might slow down, and another enzyme, asparagine synthetase, ramps up. This favors making asparagine, which is better for just storing or transporting that nitrogen until conditions improve.
Clever resource management, balancing carbon and nitrogen.
Very much so. And, it's worth remembering what the material points out. Plants are amazing chemists. Unlike us animals who need certain essential amino acids in our diet, plants can synthesize all 20 standard protein amino acids from scratch, using nitrogen from assimilation and carbon backbones from photosynthesis and respiration.
Which leads to that herbicide connection you mentioned earlier, targeting plant-specific pathways.
Exactly. Some herbicides, like glyphosate, work by blocking enzymes and amino acid synthesis pathways that exist in plants but not in animals. That's a direct application of understanding this fundamental biochemistry.
Fascinating. Okay, let's shift gears to that other major energy consumer, biological nitrogen fixation.
Right. Breaking that N-triple bond N molecule directly from the air. The material emphasizes this is the main natural pathway for new nitrogen to enter ecosystems.
And it's done by bacteria.
Yes, certain bacteria and also some archaea possess the necessary enzyme system. Some live freely in soil or water, but for plants, the really significant input often comes from symbiotic relationships.
The classic example being legumes, peas, beans, clover, partnering with rhizobia bacteria.
That's the most well-studied, definitely. The bacteria live inside special structures the plant forms on its roots, called nodules.
But not just legumes. The material mentioned others.
Correct. There are actin or hysal plants, like alder trees, that partner with Frankia bacteria. Some ferns partner with cyanobacteria. Even some grasses form associations. It's a surprisingly widespread strategy.
And the big challenge for these nitrogen-fixing bacteria is oxygen.
Oxygen, yes. The enzyme responsible, nitrogenase, is extremely sensitive to oxygen. It gets irreversibly damaged.
So how do they do this job in an oxygen-rich world?
They need microanaerobic or anaerobic conditions right where fixation is happening. Cyanobacteria might use specialized cells called heterocysts that lack oxygen-involving photosynthesis. Free-living aerobic fixers might respire very rapidly to consume oxygen quickly. And in the legume symbiosis, the plant plays a huge role.
The nodule structure itself.
Exactly. The nodule is specifically structured to limit oxygen diffusion to the interior where the bacteria reside. And crucially, the plant produces a unique protein called legumoglobin.
Legum. Hemoglobin, like our hemoglobin.
Sort of. It's related. It binds oxygen very effectively. Its job is to deliver oxygen to the respiring bacteria. They still need some oxygen for energy, but keep the concentration of free oxygen extremely low, protecting the nitrogenase. It's why active nodules often look pinkish inside.
Amazing adaptation. How does this whole partnership even begin? Plant meets bacterium.
It's a highly specific molecular dialogue. It typically starts when the plant is nitrogen limited. The roots release chemical signals, often flavonoids.
Attracting the right bacteria.
Yes. If compatible rhizobia are present, these plant signals switch on bacterial genes, called nod genes. This leads to the bacteria producing their own signal molecules.
The non-factors.
Precisely. Non-factors are lipochaetoligosaccharides. Their specific chemical structure is recognized by receptor proteins on the plant's root hairs.
So it's like a chemical handshake? Plant signal, bacterial response signal.
A very specific handshake. Binding of the correct nod factor triggers a signaling cascade inside the plant root cell involving things like calcium spiking. This tells the plant, okay, the right partner is here, let's initiate nodulation.
And then the physical process starts. Nodule formation.
Right. The nod factors cause the root hair to curl around the bacteria. An infection thread, basically an internal tube made of plant membrane, forms and grows inwards, carrying the bacteria into the root cortex.
While the nodule itself is developing.
Simultaneously, cells in the cortex, prompted by plant hormones influenced by the nod factor signaling, start dividing to form the nodule primordium. The infection thread grows towards this, then releases the bacteria inside the plant cells.
But the bacteria stay contained.
Yes, they're enclosed within a plant-derived membrane forming the symbiosome. Inside this, the bacteria multiply and then differentiate into the nitrogen-fixing form, called bacteroids. The whole nodule develops vascular connections to trade nutrients, sugars from plant to bacteria, fix nitrogen from bacteria to plant.
And inside those bacteroids, the nitrogenase enzyme is doing its work. N2 plus a lot of energy.
A lot of energy. The reaction is N2 plus electrons plus protons plus 16 ATP, yielding ammonia, NH3, hydrogen gas, H2, and 16 ADP plus phosphate.
16 ATPs for N2 molecule. That's a huge biological cost.
It's one of the most energy-expensive biochemical reactions known. And nitrogenase itself is quite slow, so the bacteroids are packed full of this enzyme. It can be up to 20% of their total protein.
And in the ammonia they make, how does the plant actually use it?
The ammonia produced by the bacteroids is quickly assimilated right there in the nodule cells, usually into amides like glutamine and asparagine, or in some tropical legumes into compounds called urades.
Okay.
These stable, nitrogen-rich compounds are then loaded into the plant's xylem, the water transport system, and shipped up to the rest of the plant to be used for growth.
Incredible system.
Yeah.
Okay, let's move away from nitrogen now and touch on sulfur assimilation. Sulfur's important too.
Oh, definitely. Essential. It's in the amino acids cysteine and methionine, critical for protein structure through disulfide bonds. It's key in iron-sulfur clusters for electron transport in photosynthesis and respiration. Plus, it's in cofactors, defense compounds, lots of roles.
And plants get it mainly as sulfate from the soil.
Mostly sulfate, yeah, absorbed by the roots. Like nitrogen, sulfate is quite oxidized, so the plant needs to reduce it to incorporate it.
Another energy investment.
Yes. First, sulfate is activated using ATP. Then, in a series of steps, mostly happening in plastids, it gets reduced down to sulfide, S2.
Sulfide.
This sulfide is then immediately incorporated into an organic molecule, usually by reacting with O-acetylserine to form the amino acid cysteine. Cysteine is the main entry point for sulfur into plant organic compounds.
And the material said this happens mostly in leaves. Why there?
Good access to the energy and reducing power like ferredoxin from photosynthesis. Also, leaves are actively making precursors like serine through photorespiration, which is needed to make that O-acetylserine. The assimilated sulfur, often as glutathione, is then exported to other parts of the plant.
Right. What about phosphate assimilation? Also absorbed from the soil.
Phosphate pi is absolutely central. Its main role, you could argue, is in energy currency ATP, adenosine triphosphate. Assimilation is largely about incorporating inorganic phosphate into ATP.
And forming other essential molecules.
Yes. Once it's in ATP, that high-energy phosphate group can be transferred to make sugar phosphates, key in metabolism, phospholipids for membranes, and nucleotides for DNA and RNA.
So the main entry point is making ATP. Where does that happen?
3 main places. Mitochondria via respiration, oxidative phosphorylation, chloroplasts via photosynthesis, photophosphorylation, and a smaller amount directly in the cytosol during glycolysis, substrate-level phosphorylation.
Got it. Okay, then cation assimilation. Things like potassium, magnesium, calcium, iron. The material said this is more about forming complexes.
Largely, yes. Unlike N and S, they aren't typically undergoing major reduction. They form non-covalent bonds with organic molecules. Either coordination bonds, where the cation links tightly to oxygen or nitrogen atoms.
Like Emmy and chlorophyll.
Exactly. Yeah. Or electrostatic bonds, where the positive charge of the cation is attracted to negative groups on molecules, like carboxyl groups on cell wall pectins attracting calcium.
And the material singled out iron acquisition as being particularly challenging. Why?
Iron's essential, but it's notoriously insoluble in most soils, especially aerobic, non-acidic soils. It exists mainly as ferric iron, Fa3+, which forms rust-like precipitates and isn't easily taken up by roots.
So plants have developed strategies.
They have. Strategy I plants, most non-grasses, pump out protons to acidify the soil around the root, making iron more soluble. They release chelating compounds that bind iron, and they have an enzyme on the root surface that reduces F3+ to the more soluble F2+ right before uptake.
And grasses do something different. Strategy 2.
Yes, grasses use a chelation strategy. They release special chelators called phytocytophores, like mignonic acid, that bind F3 plus very strongly. Then the whole iron cytophore complex is transported into the root cell by a specific transporter. Very efficient.
Clever solutions. Once inside, what happens to the iron?
It's usually kept bound to chelators like citrate or nicotinamide for transport within the plant. A major assimilation step, especially in leaves, is inserting it into the porphyrin ring to make heme. Heme is vital for cytochromes involved in electron transport. Iron is also crucial for those iron-sulfur clusters we mentioned with sulfur.
And storage. Free iron is bad, right?
Yeah, very bad. It can generate damaging reactive oxygen species. So plants store excess iron safely inside a protein shell called ferritin. The material even notes the potential for ferritin in crops like soybeans to be a source of dietary iron for us.
Interesting connection again. And finally, oxygen assimilation. Plants make O2, but they use it too.
They definitely use it. The biggest use, about 90%, is for respiration burning sugars to release energy, just like animals do.
But they also incorporate oxygen atoms directly.
Yes. Water is one source of oxygen atoms incorporated, and then there are enzymes called oxygenases. The most famous is Rubisco, the CO2-fixing enzyme in photosynthesis. It can sometimes react with oxygen instead of CO2, initiating photorespiration. That directly incorporates molecular oxygen into an organic compound.
Right. Okay, we've covered the individual nutrients. Let's circle back to the energy costs, the energetics of nutrient assimilation, which the material really stressed.
It's a major theme. Converting stable, oxidized inorganic forms like nitrate or sulfate into reduced, energy-rich organic forms takes a huge amount of metabolic energy.
That figure for nitrate, 25% of the plant's energy budget.
Something like that, yeah. For roots and shoots combined, it can be around a quarter of the total energy derived from respiration or photosynthesis, just for nitrogen. It underscores how vital but costly these processes are.
And in leaves, a lot of that energy comes directly from sunlight via photosynthesis.
That's right. Many assimilation reactions, particularly for nitrogen and sulfur, occur right in the chloroplasts. They tap directly into the ATP and especially the reducing power, NADPH and ferredoxin, generated by the light reactions. This is called photoassimilation.
When does that happen most?
Typically when photosynthesis is running strong highlight, maybe when CO2 uptake is limited and there's sort of an excess of photosynthetic energy available beyond what's needed just for fixing carbon.
Which brings us to that really interesting link the material made, the effect of CO2 levels on nitrate assimilation, especially in C3 plants. High CO2 inhibits it.
It seems paradoxical, doesn't it? But yes, in C3 plants like wheat or rice, elevated CO2 levels tend to decrease the rate of nitrate assimilation happening in the shoots.
Why is that?
There are a few interactive reasons. High CO2 means Rubisco fixes more carbon and does less oxygenation, so photorespiration decreases. And photorespiration actually helps provide some of the reducing power needed for nitrate reduction in the cytosol.
So less photorespiration means less power for nitrate reduction.
That seems to be a key part of it for C3 plants, yes. Plus, there might be direct competition for the reducing power generated by photosynthesis between CO2 fixation and nitrate reduction.
But C4 plants, like corn, are different.
They are. Their carbon fixation pathway inherently generates plenty of reducing power, as MELate than NADH, in the cytosol of their mesophyll cells where they do their nitrate assimilation. So their shoot nitrate assimilation isn't really inhibited by high CO2 in the same way.
And this has real-world implications, especially with rising atmospheric CO2.
It really does. The material flags this as a potential issue for food quality. If C3 crops assimilate less nitrogen in their shoots under future high CO2 conditions, that could translate to lower protein content in grains, for example.
A significant concern for nutrition.
Definitely. And the source suggests that one potential adaptation strategy could be breeding crops that rely more on assimilating nitrogen in their roots, or perhaps using ammonium as a nitrogen source, as root processes are less directly affected by atmospheric CO2 levels.
That's a really important connection, from basic biochemistry right up to global food security. Wow, we've covered a tremendous amount of ground here.
We really have. We've journeyed through how plants acquire and transform the essential inorganic nutrients, nitrogen, sulfur, phosphate, cations like iron, even oxygen. We've looked at the physiological processes, the energy demands, complex enzyme reactions, specialized structures like root nodules, the molecular signals involved.
Right, the nod factors, gene regulation.
Exactly. We touched on experimental examples mentioned, important terms, and tried to link it all back to real-world contexts like fertilizers, herbicides, acid rain, and food quality. I think we've managed to summarize the key aspects across the entire chapter material provided.
Absolutely. It just paints this picture of the plant as this incredibly dynamic chemical factory. So maybe the next time you look at a plant, think beyond just the sunlight and water. Consider that immense biochemical effort going on inside, the huge energy cost involved in taking simple elements from the air and soil and building the very foundation of the food web. It leaves you wondering, doesn't it? As our planet's atmosphere changes, particularly with rising CO2, how might these absolutely fundamental assimilation processes need to adapt? And what could that mean for our ecosystems and ultimately our future food supply? Definitely something to think about.